The overall goal of this procedure is to induce highly efficient dopaminergic neuron differentiation in vitro. This is accomplished by first preparing a single-cell adhesion culture of human pluripotent stem cells. In the second step, the cell culture media is switched to neuron induction media to induce floor plate progenitors. Next, the dopaminergic neurons are cultured in maturation media, and then, finally, the single-cell dopaminergic neuron progenitors are cultured on poly-L-ornithine laminin fibronectin plates in maturation media for 30 days. Ultimately, immunofluorescence microscopy can be used to assess the expression of important dopaminergic neuron markers.
This technique has a great advantage over the existing neurosphere or neurorosette protocol. It can generate a more homogeneous dopaminergic neuron population with higher efficiency.
Four days after passage, aspirate the medium from the cell cultures. Detach the cells with 1 milliliter of Accutase per well and incubate them at 37 degrees Celsius for 5 minutes. During the incubation, add 1 milliliter of gelatin to each well of a 6-well plate, and then place the plate along with freshly prepared Matrigel-coated plates into the incubator. When the cells have become semi-floating, pipette them up and down several times to make a single-cell suspension. Then pour the cells into a 15 milliliter tube and centrifuge them for 3 minutes at 250